The question is, patients that have a selective IgA deficiency can have normal levels of IgA in the serum, or actually elevated, but low or absent IgA in mucosal sites. And the question is, why would this happen? I should say the exact mechanism is currently unknown. It's actually, there are several mutations that are known. But the first is, it's not production of IgA. Okay, so you do have cells that are producing IgA, it's in the serum. You could argue that it might be a problem with the J chain, that it's not folding correctly. But in most patients, you do have dimeric IgA in the serum. The more common problem that is associated with selective IgA deficiency is actually binding to the poly-IgA receptor, right? This is the protein that will bind to IgA and transport it across the mucosal epithelium, okay? So it binds, actually is binding to the J chain, it's being transported across, but then you could have, there are known issues with the transcytosis and also enzymatic cleavage of the poly-IgA receptor, because remember, part of it becomes this secretory component that is attached permanently to the IgA and keeps it stable in the mucosal surfaces. Okay, so if are there any questions on that? Right, so today we're going to start talking about B cell development, and in particular, we're going to cover several times today somatic recombination, which is not as difficult as it sounds, but it is a necessary step for B cell development. And so if we go back to the clonal selection theory that was proposed by Burnett and Talmage in particular, was that in the naive B cell or the naive lymphocyte population, every B cell has a unique receptor, and only the B cells that are specific for some antigen are going to proliferate and respond and start making antibodies. Now the problem with that is there are trillions of potential different things that could be trying to infect you. You only have between 20 and 40,000 genes, depending on whose estimate you take into account. So you can't have one gene for every different pathogen, right? That would be far too few to actually encode the number of different antibodies to all of the potential epitopes recognized by B cells for all the different pathogens in your environment. And in fact, it may not even be enough for just even a select few. And so the immune system has developed a way to deal with this. And the way it deals with it is something called somatic, recombination. This is where a B cell will take its DNA and intentionally rearrange it in a sort of sloppy way to generate a large number of unique B cell receptors from a fairly limited set of gene segments. And we'll go actually into the numbers of how many different ones you can get. So a key takeaway from this is that a germline DNA sequence, if you look at a hematopoietic stem cell or even an epithelial cell or a hepatocyte, you look at the BCR gene locus in those cells, it does not encode for a B cell receptor. It only occurs in B cells that have rearranged their DNA to generate this new B cell receptor sequence. And it will do this for both the heavy chain and the light chains, the kappa and lambda light chains. So this was first really recognized by a guy named Susumu Tanagawa, who got the Nobel Prize in 1987 for his work that showed that B cells rearrange their DNA. And he did this by a very simple southern blot. So if you remember from biochemistry, southern blot is where you chop up the DNA and then you use a radiolabeled probe to see how fast it migrates in an agarose gel. Okay, so what he saw was that in the germline DNA, if he used radioactive probes for the constant region or the variable region of the B cell receptor, he got in a certain migration pattern. But if he looked in mature B cells, he saw that that migration pattern changed. Okay, so this is DNA that he's taken and chopped up with an enzyme with a restriction fragment or a restriction enzyme to get different fragments. And his conclusion from this was that the B cells are rearranging their DNA. What it looks like is this. In the germline DNA, you have the V regions and the C regions are separated by large segments of DNA. And in that DNA are restriction sites. So there's restriction sites on the outside and restriction sites on the inside. And so this would cut this DNA into a short piece and a long piece. But once he looked in B cells, essentially what's happened is the V regions and the C regions are much closer together, and you've lost this extra restriction enzyme site. And so now you only get one fragment. And when you probe that, it's much smaller, or it's actually not much smaller. It's a different length than either of the two that started. So this very simple experiment showed that B cells are intentionally rearranging their DNA. And so if you think about this conceptually, somatic recombination is where you take these gene segments and you intentionally stick them together in different combinations to get a fully functional or fully encoded B cell receptor. So here is our germline DNA. There's no B cell receptor here because you haven't recombined these. For the heavy chain, there are two recombination steps. One, to join the diversity and J segments, which we'll talk about in a second. And two, to join the variable region gene segments with the joined DJ segment. For the light chain, they don't have diversity segments, so there's only one recombination step where you're joining V and J segments. Now that gets transcribed as a mRNA, and then the exons are spliced out, leaving you with just, sorry, the introns are spliced out, I'm sorry, it's early morning, leaving you with the full coding sequence. Okay, now when that's transcribed, that gives you, in this case, the heavy chain, or in this case, the light chain. But I want you to notice where the joints are, because this will become important a little bit later on. This joint is actually the tip of the actual protein. Remember, these domains are the immunoglobulin top coat that's folded over and stapled with a cysteine-cysteine bond. And that is then it's just the fingers that are at the tip of that fold that are the antigen recognition part. So the joints of these are at the tip of that domain.
Sorry, what about the C region for the heavy chain? Where does that?
It's not recombined. You just have those, each of those is one immunoglobulin domain. So this domain encodes this, and because it's constant, all IgM have the same constant regions.
Sorry, I meant the purple region. Where does that?
That's actually the hinge region. That's this part right here. They don't typically draw it in purple, but that's the hinge. Okay, so if we look at the human B cell receptor loci, you have in your genome about 70 or so V gene segments for the lambda and about 7 to 11 J segments. Now in the lambda, its HJ is paired with a C region. All of the C regions are the same. It's just those arose by gene duplication. So they're you can have C lambda 1 or C lambda 2, but they're all the same. They have the same sequence. And so there's no difference in the function of those. But the J's have a different sequence. And so what you're trying to pair is a V to a J, and along with that comes a C region. For the kappa, it's a little different. You only have one C kappa region, but in front of that, you have about 5 J segments. Now there's more variation in the number of V gene segments you have, and I don't care that you know exactly how many V's or J's there are. in general, there's far fewer J's than there are V's. But in this case, you're going to join a V to a J, and then whichever the C region comes along with that, no matter which one you choose. The heavy chain has a bit more diversity. It has somewhere in the order of 120 V gene segments, and then 9 J segments. And then there are, in humans, about 27 D gene segments. Okay, so there's a lot of different choices here, but if you were just to choose, that's still not quite a lot of diversity. And you can actually do the math on here, right? Now, I will say that this is drawn a little bit incorrectly, right? Then we know that this C region for lambda, or this C region for kappa, for the light chain, that's one IG domain. When they draw this, typically people will just put one box for C mu, which encodes the IgM constant domain, or one box for C delta, which is the IgD constant domain. In reality, remember, these are actually, if you go back to the previous slide, the IgM is actually 1, 2, 3, and it has a fourth one, has 4 constant domains. So technically, these should be drawn as 4 different boxes plus a hinge region. But in any case, it just gets cumbersome to try and draw all of those. Now, when you do the math on this, if we only take into account just VDJ gene segment selection, how many possibilities could you have? Well, the first is, if for just the heavy chain, just the number of different possible combinations gives you about 31,000 different things that you can recognize. Now, that's a huge step up from, if you've only got 20,000 genes, you're already more than the entire genome in possible things you can recognize. But that has to pair with a light chain. And so if you look at the possibilities for kappa or lambda, you have about 1,000. And so when you multiply those together, because remember, binding is both the CDR1, 2, and 3 from the heavy chain and the CDR1, 2, and 3 from the light chain, you get about 37 million different possible combinations. And that seems like a lot, but compared to the trillions or higher numbers of pathogens or potential things on the planet that you could recognize, it's still not high enough. And so what you have to do is then use an evolutionarily sloppy way of joining these together to get sort of an increase in diversity. And we'll talk a lot about that today. Okay, so we talked a little bit about where that joint occurs between the V to J or the V to D to J segments for the light chain and heavy chain respectively. And that is at the tip, right? It's part of that, the fingers extending from the immunoglobulin taco that form the actual antigen binding site. Now, there's 2 different kinds of CDR or HVR1 and 2, okay? HVR1 and 2 are encoded by whichever V gene segment that B cell chose. So all heavy chains that used the V heavy 13 gene segment have an identical HVR1 and HVR2. Okay, so that's just determined by which gene segment you chose. The HVR3 or the CDR3, and in this class we're not going to really differentiate between the two. Remember, they came about from different methods, but the CDR3 is the joint between the V to J for the light chain or the joint between the V to D to J for the heavy chain. So 2 B cell clones that used VH13 will have the same HVR1 and HVR2, but a different HVR3. Okay. And so when you look at this, once you have this joint made, the CDR1 and 2, which correspond to the HVR1 and 2, those are just the whatever gene segment, right? And they are variable, but they're variable between gene segments. The CDR3 is the joint. And so when you look at Here's our folded immunoglobulin taco. All of the yellow parts are the beta-pleated sheet that's been folded over on itself. You can see the disulfide bond here. The HVR1 and 2 are less diverse than the HVR3, but they're at the fingertips that are extending off of that immunoglobulin fold. The HVR3, which is typically much more diverse because it's this random joint that's put together in a sloppy way, that's also one of the fingers that's extending from the IG fold. So the CDR3, or HVR3, their definition is fine, is the most diverse portion of the antibody. And if you recall from last lecture, when you do structure studies of what part of the antibody is binding to the antigen or the epitope, the CDR3 makes the most extensive contacts. The CDR1 and 2 also do make contacts, but the CDR3 is usually the part that really determines what the antibody is specific for. It's saying this is the most diverse part. Okay, but an important concept is if you have a B cell clone and you grow that up, all of those B cell clones have the same CDR1, 2, and 3, or HVR1, 2, and 3 sequences, because they originated from the same mature B cell that after the somatic recombination has occurred. So how does this work? Well, if you're going to have a system for recombining DNA, you can imagine that that is fraught with perils in terms of generating cancer. For example, taking a strong promoter and putting it in front of a proto-oncogene will give you a tumor cell. Okay, so that you don't want this to happen just random in the DNA. And so this is controlled by the RSSs, which are recombination signal sequences. And these are just stretches of DNA, but they have a particular structure, and there's two different kinds. One is the 7 nucleotide spacer and 9 nucleotide segment, in which the spacer is 23 nucleotides. The other version is a 9 nucleotide, 12 nucleotide spacer, and 7 nucleotide. Okay, now we typically just write them as a 12 or a 3, but in reality, these different parts have very important roles. The first is that the 7 nucleotide sequence, which is palindromic, is the same on both ends, okay? And that's really important for formation of or pairing these RSSs together, okay? The 9 nucleotide is that the sequence is important for making sure that they're actually going to recombine or they're going to get together. Now, the importance of the spacers comes about in that you're pairing up the 9-mer with the other 9-mer. You're pairing up the 7-mer with the other 7-mer. And so what happens? Well, the 23 nucleotide spacer gets bulged out, sort of gets looped out to the side. And this turns out to be what is recognized by the recombination machinery, so that you can see there's this non-identical DNA. It's bulged out on one side. And so the rule is that you can only have recombination between a gene segment that has a 23 nucleotide segment or a 23 nucleotide sequence spacer with a gene segment that has a 12 nucleotide spacer. So if we go back and look, sort of just did a cartoon form, for the lambda chain, the V gene segments have a 23, and the J gene segments have a 12. Now we've only drawn one here, but they're all the V's have a 23 and all the J's have a 12. For the kappa, it's just the opposite, right? The V's have a 12 and the J's have a 23. Now for the heavy chain, it's a little more complicated because you have first D to J recombination and then V to D to J recombination. Now which happens first? It's first D to J. So here we have a 12 and a 23. And then once that's recombined, you still have a 12 on this side and a 23 on this side. The question is, can you get a V to J recombination for the heavy chain? Well, no, because here you have a 23 and a 23, so it wouldn't work. Okay, so this is one of the important parts of immunology, is controlling how the gene or how the DNA is recombined. And so this 12-23 rule is you can only get recombination between 23 nucleotide spacer RSSs and 12 nucleotide spacer RSSs. Now, somatic recombination, which we're going to cover today for B cell receptors, we're also going to cover it on Tuesday for T cell receptors, is a regulated process. And it's essentially the same whether it's for the heavy chain or the light chain. The same processes happen. And we'll talk about how many times this is going to happen for a developing B cell. The only difference is the timing of when this happens. The heavy chain rearranges first, and then there's a step, and then the B cell is going to rearrange. And we'll tie in somatic recombination to those actual steps of B cell development. Okay, but it's the same process. One of the unique parts of the immune system is that you have proteins expressed in these developing B cells that are not expressed in other cells. And part of that is because in other cells, you don't want DNA repair to be sloppy. In developing B cells, you do. And so a lot of the proteins that are involved in somatic recombination are just normal DNA repair proteins. But there's a few that are lymphocyte specific. And these include the RAG1 and 2 genes, that stands for recombination activating gene. TDT, which is terminal deoxynucleotide transferase, which is a unique nucleoside polymerase that works in a different way than any of the other polymerases you've encountered. And a protein called Artemis, which has a unique role in the immune system. So if we just take the steps here of what happens during recombination, you have a developing B cell and you express these RAG proteins. In reality, RAG1 is determining which V gene segment or which gene segment is picked. RAG2 is the enzyme that actually cuts it. So the RAG proteins bind to the 12 and 23 RSSs, and then they bring those together. So that you're pairing up the 7-mer and the 9-mer so that you're making sure they fit. And then RAG2 is recognizing that bulge and is cutting the DNA. Okay, so that's really all the job of the RAG proteins. Now, in your normal DNA repair process, you have these proteins called Ku70 and Ku80, and they form a dimer, and their job is to bind double-stranded ends of DNA. Basically, any DNA that gets cut up, these proteins come in and they stabilize it. So there's really two things that happen. If you look, there's two parts, right? There's the part you've cut off, and then the part of the DNA that you want to retain. And so we'll talk about those two differently. But typically, the part that's cut off is called the signal joint. That's just the part that had the RSSs on it and was recognized to signal this is what you're going to recombine. The coding joint is what's actually going to encode the B cell receptor.
Dr. Blattman,
Yes.
quick question. So the Ku7080 is not a lymphocyte unique protein, it's a separate one.
Yeah, it's just a normal DNA repair enzyme that's expressed in many cells.
Okay, all right, thank you.
Okay, so the first thing is,
Dr. Blattman,
yes.
I have a question about the RSS regions that get cleaved off. What happens to them afterwards? On the next, yeah, here.
Yeah, that's what we're going to talk about right now.
Oh.
Because that is actually still sort of an unknown in immunology, but this is sort of junk DNA. You don't need it anymore. Excuse me, just a second. All right, a little froggy this morning. So you're cutting out this large section of DNA between the V and J segments, or the other gene segments, and can be quite large. And so what do you want to do with it? Well, having DNA that has sort of free DNA ends that's just cleaved is a bad idea for a cell, right? Because those can be then incorporated back into DNA. They can be inserted in different places, and so you don't want that. What you would like to do is that make that into a circular piece of DNA that's not going to recombine very easily, and that eventually you can just take care of and degrade. And so what happens is the Ku7080 proteins actually bring the ends together, and a DNA ligase comes in and seals the break. And so what you end up with is an excision circle. Okay, so it's just similar to plasmid DNA, where you just have a circle of DNA. It's in the cytoplasm, and eventually nucleases come and degrade it. Now, you can actually use this to identify recently matured B cells that have just come out of the bone marrow, because they have excision circles either for the heavy and light chain, and they have not yet degraded them. So that's one way to identify, like, this is a new B cell. And it's sort of, this has given us estimates of what's the output number of B cells, how fast are we developing B cell number or B cells outside of the bone marrow. There's a couple of exceptions to this. There are a few V gene segments that are inverted. There's not that many, but there's a couple. And so for the normal case, if they are in the correct orientation, or the normal orientation, we'll call it, where the 23 RSS is facing the 12 RSS, then you get an excision circle. For the rare V gene segments that are in the opposite orientation, where the RSS is pointing away or pointing away from the J gene segments, you actually get a gene inversion, but it just results, instead of an excision circle, you get sort of a switch in where the gene segments are in the DNA. For this class, I'm mostly interested that you know there's an excision circle, okay, that this is actually cut out of the DNA, because that's the normal situation. Now the coding joint, on the other hand, has a different story, right? This is the actual parts that are going to encode the B cell receptor. Here's our leader segment, here's our V gene segment, and our J gene segment. And those are still bound by the Ku7080 proteins. Now, one difference here, which is a little bit hard to see, is that these are not free ends, right? Free ends would just be sticking off like this. The ends here actually are looped back on themselves. And it's not clear that this is enzymatic. It looks like this is a free oxygen attack on the other end that just occurs naturally. Some people would argue it's actually enzymatic, that there's another enzyme that does it. Right now, we don't know, so I'm not going to make you know whether that is true or not. But this is an important step because it is the first step of making a very sloppy joint, okay? And I'm not meaning that in a Snoop Dogg way. This is making the coding sequence actually some random, inserting some randomness into the coding sequence. And so the job of Artemis is to come in and cleave that hairpin at some random site. And so if it cleaves it right in the middle, you end up with two ends of the same length. But it can also cleave it highly biased on one side or the other, and so it's a non-normal length. Okay. Now there's two proteins here. There's DNA PKCS, that is a DNA protein kinase. That is the activator protein. That phosphorylates Artemis, and Artemis is the protein that cleaves that hairpin. So this is the first step where you actually get some randomness in the DNA sequence. Where Artemis cleaves is the first random part. The second step is, now you have this enzyme called terminal deoxynucleotide transferase. The name should tell you what it does. It is taking the end of the DNA and adding random nucleotides to that DNA. And so you're just getting some random sequence there that is being inserted. So these are the two main steps for generating additional diversity in a B cell receptor. One is, where does Artemis cleave? And two, how many different nucleotide combinations can you add? Now, obviously, if you're adding nucleotides, sometimes you're going to add too much. And so there's an exonuclease 1 that kind of comes back and trims back some of the ends that TDT has generated. But essentially what you end up with is some complementary sequences. DNA repair enzymes come and fill those nucleotides, and then DNA ligase seals the coding joint. And we're going to go over this again because this is sort of the high level view of it, just looking at DNA as these little strands. But when you actually look at the sequence level, it looks a little different. Okay, so what the take-home message from this part is that if you have the signal joint or the signal sequence, that has a precise joint. You're absolutely cutting right at the RSS, at the 7-mer of the RSS. For the coding sequence, you have an imprecise joint, meaning you've added nucleotides, you've made it loop over and cut it at a random place. So it makes it random in between the joint between a V to J. So when you look at these together, this is sort of, if you were looking at the nucleotide level, this is just for the coding sequences. You get the RSSs are brought together. The RAG proteins are going to cleave these off. So here it would cleave here. Here it would cleave here. And you generate these hairpins where the DNA is looped over on itself. So the first thing that's happening is that Artemis is going to come in and cleave that. Okay? And in this case, it cleaved it right at the end of the D gene segment and right at the beginning of the J gene segment. Okay? Now, that's not all, right? TDT is going to add nucleotides to the ends of that DNA. And so what happens is that you'll actually get some what's called micropairing. So here, this T will recognize this A, this C will recognize that G, but then you have nucleotides that are sitting over the edge. And so exonuclease 1 will come in and trim those off. Then you have DNA polymerase will just come in and fill those gaps. And then you have DNA ligase comes in and seals the gaps. Okay? And so what that results in are two sets of nucleotides at the joint. And they're called P nucleotides and N nucleotides. And the P nucleotides are determined by where Artemis cleaves. Because it's a hairpin, right, it's folded over on itself, wherever it cuts, it's actually going to form a palindrome, right? TCGA, if you were to do the reverse complement, would be TCGA. And so you get a palindrome from wherever Artemis cut. Those are your P nucleotides or palindrome nucleotides. The ones in the middle are there because TDT added those. And so they're non-templated nucleotides or the N nucleotides. Okay, so you have palindrome sequences and then non-templated nucleotide sequences. All right, so we're going to go through this one more time, and I've sort of tried to integrate sort of the macro view and the micro view of this so that it's a little easier to understand. And here are really only our 6 groups of enzymes, right? We have RAG1 and 2, which are the parts that are going to cut, Ku7080 proteins, DNA ligase, the exonuclease 1, TDT, and then our DNA PKCS and Artemis. So the first question is, here's our gene segments. What are the first proteins to come in?
You have RAG1 and RAG2 bringing in the 12 and 23 portions of J and V.
Right. So which one determines which V gene segment you're going to use, RAG1 or RAG2?
Good question.
It should be RAG1.
Right, RAG1 gets to choose. Now normally we do this exercise in class. I bring in this long thing of yarn and then I ask for volunteers to come up and they have to actually cut the yarn and tell me where they're cutting it. But since we can't do that with social distancing, we're going to do that virtually. So the first thing to come in are the RAG1 and RAG2. Now in reality, it would be a little cluttered if I put everything on here. There would be a RAG1 and RAG2 over here too. And they're bringing those 12 and 23 RSSs together. It looks like something like this, okay? So Rag1 got to choose which gene segments we're going to use. Rag2 will cut. Where does it cut? Before the RSS or after the RSS? Or does it cut close to the V gene segment or distal from the RSS? For example, does it cut here or does it cut up here?
Next to the V gene.
Right, it cuts right here. It's a precise cut right after that heptamer because you need to control this so that there's not some aberrant thing there.
So it's distal to the.
Well, no, it's proximal to the gene
Proximal.
segment, right? It's right next to it. Okay, so now we've got two sets of DNA. We've got a DNA that's got the RSSs with the RAG1 and 2 proteins. Those have free ends. And then we've got the coding sequences that are not free ends, they're hairpins. What's the next protein to come in?
KU7080.
Okay, so KU7080 comes in here. It also comes in here. So that's step 3. What's the next step? Let's talk about for the excision circle. What happens? What's the next protein?
Phosphorylated.
What's already phosphorylated from being cut?
Doesn't DNA ligase close it?
Right. DNA ligase 4 comes in, seals the gap, and you're done. Okay? And then that can just essentially is in the cytoplasm that circle will eventually get degraded. So it's just a circular piece of DNA.
Really quickly, how long does that circle, how long is it present in the cell, like after B cell development?
I don't know the current estimates of how long it's there. I think you can measure recent B cell immigrants up to a couple weeks. They'll still have it. But it doesn't
Wow.
last in the cytoplasm for very long because you can imagine it's dangerous to keep DNA around, and it would be beneficial to recycle those nucleotides and use them for other things. Okay, now what about the coding sequence? What's the next thing that happens, or what's the next protein to come in?
Artemis?
Artemis. And what activates Artemis? It has to be phosphorylated by DNA protein kinase CS, right? CS is for class switch. So this is, what does Artemis do?
It's going to cut randomly.
Okay, so it's going to see this stem loop or this hairpin and just cut it at some random sequence. Now, it's easiest to think of this if it's a sort of asymmetric cut, right? So you have this part of it is much longer than this part. What are these nucleotides called? Are they P or N nucleotides?
P.
These are P, that's the palindrome. Because it was a hairpin, you're going to get the same sequence, right? The complementary sequence is going to fold out. Oops. Okay, so Artemis has cut this. That's the first step in generating some junctional diversity. What's the next enzyme to come in?
TDT.
Okay, so TDT comes in, and what does it do?
It's going to add more the non-template nucleotides.
I hope you all appreciate my animation skills here. Right? So it adds more nucleotides. Is it taking them from a template?
No,
Right. So what are these nucleotides called? Are they P or N?
the N.
They're the N. They're the non-templated nucleotides. What's the next thing that happens?
Exonuclease comes in.
There's one small step in between the exonuclease coming in and TDT. And that's really these micro pairing, right? Where you're getting some nucleotides sticking together, and then you have these parts that are overhanging. So exonuclease is really coming in to chew off those overhangs. Okay? Now what happens? We've
Is
got a
this where
little
you
bit
can
of,
get variability in antibody, like the number of amino acids in a single antibody? Because
correct. The number of nucleotides you add or chew away determines how long that CDR3 is. And we'll come back to that question in just a second. But yes, that's where you get this difference in length. So what do you, excuse me, what do you have to do next?
Ligase.
So you actually have polymerases that fill in these gaps, and then DNA ligase will seal those gaps. Now, we don't talk about the polymerases because it gets a little complicated. There's different polymerases for the heavy chain and the light chain. Nobody really understands why, but there are. And so just know that you do have to fill in the missing nucleotides because DNA ligase is just a ligase, it's not a polymerase. Okay, and now you have this complete sequence. This is a complete V to J sequence. Now the question is, how does a B cell know that you've done this? How does it know that you've completed this task? Well, there's a couple of issues, right? One is that all of these loci are on separate chromosomes. The heavy chain is on chromosome 14, kappa is on 2, lambda is on 22. So you have to have some control over making sure that you have only one B cell receptor per B cell, and also that you're not recombining everything over the same time. And so in order for us to sort of maintain the clonal selection theory that each B cell expresses one B cell receptor sequence, and during a response, only B cells that have the correct sequence will respond, you have to have some mechanism for ensuring that you have only one heavy chain or only one light chain per cell. And this is often called allelic exclusion. There are other terms for it. But the problem is because these are all different chromosomes and you have 2 copies of each loci per cell, you actually have to have a system for testing, did I get a complete sequence? Okay, and we'll come back to that when we go through B cell development. Okay, and so the idea here is that you've got to maintain control over this. And the way that you do this is you only do one at a time. Okay, so this is really where we tie into B cell development. How does a B cell control that recombination so that it forms a new B cell with a new B cell receptor and it doesn't recombine the DNA in other ways? So this occurs in a couple of steps. The first is the developing B cell will rearrange the heavy chain first. Any ideas why? Think about how many different gene segments you have to rearrange for the heavy chain versus the light chain.
It's more complicated, so
Yeah, it's more complicated. So you have to do that one first. If you get that rearrangement to work, the light chain's much easier. Okay, so you rearrange the heavy chain first. But the common theme in both B cell and T cell development is you rearrange something and then you test if it gets expressed. If it is expressed, yet you get to go on in the development process. If not, you go back and try it again. Okay, so this is just a logic thing. If you rearrange it, you test for expression. If it's there, yes, great, keep going. And again, the light chain then rearranges after the heavy chain. And it's not clear, there's a huge bias between whether kappa or lambda is used in a different species, and it's not clear why, but there is a huge species bias for which one you prefer to use.
sorry, what are the tests for the
We'll come back to
rearrangement?
the test in a second.
Oh, okay.
Because we're going to tie it into the steps of B cell development it's got to do this rearrangement at a certain step and then test at a certain step. And so we'll go to that in a second. Okay, so again, you test whether or not you got rearrangement. And then the last step is, okay, if you did get a productive heavy chain and a productive light chain rearrangement, then you got to make sure self-reactive B cells get deleted, okay? Now, at all of these steps, if you look back, if you don't pass the test, you either get to go back one step or die. If you have self-reactivity, would you get to go back all the way to the heavy chain rearrangement? No, you only get to go back one step. So if you fail this step, you get to go back one. You fail this step, you go back one. In this case, if you fail this step, you're actually going back to the light chain rearrangement. Okay? So let's talk about what happens at each of these steps, okay? Now, we don't go into all of the signals, and here I've included some surface markers for how you can tell which of these cells is which. But really, we have to go back to the hematopoietic pathway, where you have a hematopoietic stem cell, it's going to give rise to a multipotent progenitor, which gives rise to a common lymphoid progenitor. So that turns out to be, for the lymphoid development pathway, B cells are the default. And so you typically have many more B cells than T cells, because to go down the T cell pathway, you require additional signals or additional molecular signals that tell it to become a T cell. B cells don't need those, so they go, if there's no other signal, it just becomes a B cell. Now the first step is the pro-B cell. Okay, and so pro-B cells are going to upregulate RAG1 and 2 proteins. Which loci is it going to rearrange? Which one goes first?
Heavy?
Right, so it's going to do the heavy chain. But remember, there's 2 steps. Okay, so we divide the pro-B stage into two different versions, the early pro-B and the late pro-B. The early pro-B, remember, for the heavy chain, it's got to do a D to J recombination first, and then it has to recombine the V gene segments with the D to J joint. So you actually go through that entire process of somatic recombination twice at the pro-B cell stage. You go through it once in an early pro-B cell stage where everything happens, you join D to J, and then you go through that entire process again for the late pro-B stage where you get V joined to D to J. Now how it's testing this, right? You've now recombined this, and now you need to test if there is actually something on the surface. The way this works is that there is an enhancer that's downstream of the J gene segment. And so it has a promoter with each of the variable region gene segments, but there's no enhancer, and so the expression is not turned on. If you have full joining of a V to D to J, that brings the promoter close enough to the enhancer. Well, this will then turn on. Okay, so now you're actually getting mRNA from this, whereas before the promoter was too far away and it wasn't working. Okay, so now once you've joined them, you get mRNA. That's then translated into protein, and you have a chance to get something on the cell surface. Right, but that's only if it's in the right reading frame. How often is it in the right reading frame? How big is a codon? I'm sure you all remember from biochemistry. A codon is how many nucleotides?
3.
3. So if you have TDT as adding random sequences, only a third of the time will it be the correct length. Right, so 33% of the time, it's going to be in frame, meaning it's not going to encode a garbage sequence that ultimately results in a stop codon or something. Okay, so you're really testing, did I get something in frame? We're not testing what it recognizes or anything like that. We're really just trying to test, did I get recombination in the right frame? That's because 2/3 of the time it's out of frame. And so what you're testing, did I get a heavy chain on the cell surface? So you can imagine if it's out of frame, you could get a stop codon. It also changes the entire reading frame downstream of it for the constant regions. So you get stop codons and it doesn't work. So if you look at the length, and this was one of the questions, is the variability in the length of that CDR1, or sorry, that CDR3, it can be quite a large distribution, but it can only be in discrete sets of 3 nucleotide differences. Okay, so each of these is how many cells have this nucleotide length, or the CDR3 length is this long. But you notice there's not any in between. And so that's because if it's 1 or 2 nucleotides more, it's out of frame, it doesn't get expressed. Okay, so only in-frame recombination is productive, and so that's the only thing that's going to get actually out to the cell surface. And what happens is you get the heavy chain. So here's our heavy chain that's being expressed. But the problem is heavy chains are not stable without their light chains. You haven't rearranged the light chain yet. So what you need is the surrogate light chain. And it's really just 2 proteins. They're soluble, they're expressed by B cells. And these bind to the newly rearranged or the newly expressed heavy chain and stabilize it. And so those are the V pre-B, which is just a protein that looks like a V region, but there's no variability in it, and the lambda 5, which is part of the Ig lambda loci, but it's a constantly expressed protein. But those are just stabilizer proteins. Now what's happening is these get expressed on the surface, and they get expressed with the signal proteins. Remember, the Ig alpha and beta are the BCR loci, or the BCR genes that are expressed that have the ITAMs, the intercellular tyrosine activation motifs, that can lead to kinase activation. And so what's happening here is the surrogate light chains are cross-linking on the surface with other surrogate light chains, and they're bringing all those together, and that allows this phosphorylation to happen. We're not going to go a lot into the signaling cascades that happen downstream of a B cell receptor, just because I think it's enough to start realizing there is a signal here. So if you get a heavy chain on the surface, then with the surrogate light chain, that gets cross-linked, and you get activation of kinases that tell the cell, yes, you got something expressed, keep going.
So is the presence of this surrogate a good marker for pro-B cells?
It in fact is the marker for pro-B cells. Right, we have other markers, but expression of the surrogate light chain defines pro-B cell stage. Okay, now let's say that you're a developing B cell. You've done this, and you've got a positive signal. And now you want to capitalize on that success. What you do is you divide several times, right? So that now you have, instead of 1 B cell progenitor that has 1 new B cell receptor, you've got several progenitors, all have the same heavy chain. But because you're now going to rearrange the light chain, now you can have lots of different specificities. You're sort of just maximizing on the success of that first step. So once you get a signal from expression of the heavy chain, that cell moves on to the pre-B cell stage. And that is defined first by the large pre-B followed by this small pre-B. So for pro-B, it's early pro, late pro. For pre-B, it's large pre, small pre. In biology, what happens when we talk about a large cell? That's often called a blasting cell, or a blast type of cell. There's no difference. B cells that are large are dividing, right? Because they've got to share cytoplasm, they've got to recreate everything and then split it between 2 cells. And so the large pre-B stage there's no recombination. It's actually just cell division. Okay, and at this point, it's losing surface expression of the heavy chain, because now You don't want it to keep getting those signals to proliferate. The second step is the small pre-B stage. And this is where you actually are rearranging the light chain. Now, we're not still really sure how you control rearrangement of kappa versus lambda. It may be stochastic. You're just looking for how, any one that works, that you take the first one that happens. But there is a bias in which one you use. And also, we don't talk about this a lot in immunology, but functionally, there could be two different light chains on the same cell. And functionally, it still is only a single specificity. And we'll talk about that when we get to B cell activation, but it's really one, if you get two different light chains, one may not actually work. It may never bind, it may never see antigen, and so it doesn't really care. Okay? Now at this point, you have to again test. Did I get a light chain rearranged? And the way that you test is saying, okay, now I have the heavy chain and I have my light chain. We've turned off the surrogate light chain, so the only way that I can have a stable expression on the surface of that B cell is that I get both the heavy and light chains expressed. Now, how does it know whether it's expressed? Well, there's something called a tonic signal, which is sort of just a very low-level kinase activation that tells the cell, yes, there is a protein on the surface. It's not clear that there's any ligand for this. It's mostly just expression levels and grouping together of the BCR on the cell surface. But it tells the cell, yes, you do actually have something here. Okay, now that says go to the next step, which the next step is, okay, we have to test whether or not this new B cell receptor that we generated formed a self-reactive BCR. Because if it does, we got to get rid of it. Okay, and so it's actually testing this by looking for proteins in the bone marrow that it can recognize. And so if it does recognize any of those, it can go back one step. But let's go back to what happens if you failed all of this, right? Let's say, yes, you got a B cell receptor on the surface first, fine, you can go on, but 1/3 of the time, that's going to happen. 2/3 of the time, you're going to get out of frame rearrangements, and so you go back one step. And so there's some rescue, and this is called B cell rescue of somatic recombination. And this is if there's no signal, what else can you do? Well, you've got several copies of the light chain, and this is probably why you have a kappa and lambda, is because you don't want to run out of options. You've spent all of this energy rearranging the heavy chain. You want to make sure that the light chain is going to be successful. And so you can rearrange this several times. So let's say that your first try, you use this gene segment and this gene segment. Well, that cut out everything in between. But there's no reason, if this didn't work, that you couldn't do this one. Now that would cut out all of this intervening segment. If that didn't work, then you can try again with this one. And the only thing that you're going to run out of is eventually you're going to run out of most likely J gene segments, right? If you run out of J's, there's nothing to recombine to, so you'd have to switch to the other loci for that same light chain or the other light chain. So you've got lots of options here. If eventually you keep doing this and you don't get a productive expression, then that cell dies. Okay, so the heavy chain you can rescue at the pro-B stage, but there's not as many options because you have to have a D and a J and a V. And so you sort of run out of options much quicker for the heavy chain. For the light chain, you have a lot more options. If you run out of options for either of them, that cell just dies. Okay, but if it does get expression, if we go back here to our heavy chain, or our heavy and light chain are both expressed on the late pro-B stage, then that provides a signal. Now you want to test, is it self-reactive? And that happens at the immature stage. And I want to point out just one thing here. These cells are positive for IgM, but they do not express IgD, okay? And that's how you differentiate between immature and mature. And it's really only cells that are going to pass through this negative selection are going to go on to become mature B cells. And so what's happening is you go from pro-B to pre-B to immature B, and you have heavy chain rearrangement to get in the pro-B stage, light chain rearrangement in the pre-B stage. You get to an immature B cell, and now you have to test. Before that B cell can leave the bone marrow, you test if it's going to recognize any self-antigens. Now, in reality, it's really only testing if it recognizes any serum proteins, because it's really proteins that are circulating in the serum that are binding to complement proteins in the bone marrow and being held there for B cells to recognize. So it's not fully deleting all self-reactive cells. It's just mainly deleting self-reactive cells that recognize anything they would normally see in their circulation between the blood and the lymph. So what happens? Well, again, if it gets a very strong signal that tells it, yep, you recognize self-proteins, go back to this previous stage and rearrange the light chain again. That's called receptor editing, or B cell receptor editing. And it keeps doing this and keeps going back to be tested until it runs out of options. And if it failed all of its options, then it just is going to die or is deleted. But there's another thing that can happen if it gets a weak self-reactive signal. It becomes anergic or undergoes anergy. Anergy is the opposite of energy. Anergy is the lack of an ability to respond. Often how many of us are feeling in this self-isolation, we just can't have any, we don't have energy, we have anergy. Okay, now it's not really clear why the immune system does this, because eventually anergic cells are going to go out and they eventually get deleted anyways. And so it's not clear if anergy is on the pathway to for deletion, but it does happen. And the only time that a B cell can actually get out of the bone marrow and go and look for antigen, do its job, is if there's no self-reactive signal. Okay, and when it does this, it now will express, in addition to IgM, it will express IgD. Remember, this is the only time in a B cell's life where it can express two different isotypes. And the reason is thought that now you're getting more surface expression of immunoglobulin or B cell receptor. And so you have IgM and IgD, and they both contribute to signaling. So now you're sure that's not a self-reactive cell, so you make it more sensitive once it leaves, and it can go out and respond to infection. Now that really defines a mature B cell. Once it expresses IgD, it's mature. How does it do that? Well, it's just alternative mRNA splicing between IgM and IgD constant domains. It's not somatic recombination, it's not isotype switching, it's just differential RNA splicing. Okay, now those cells that got no signal during negative selection will exit to the blood, and they come out into the blood via the central sinus vein that's serving that bone marrow. So it's just now it's going to go in the blood, then it's going to enter a lymph node via the high endothelial venule. I want to point out a couple of other things here that maybe were not quite clear. If we go back to complement receptors, CD19 is part of complement receptor 2. And so it starts to express this, but really it's trying to look for antigen at this immature B cell stage to select against self-reactive cells. Remember, CD19 is part of the B cell co-receptor. We go back to the last lecture, we talk about the BCR complex, CD19 is part of that, right? It's one of the proteins there, and its job is to recognize proteins that are tagged with IC3B. And so these are being held by complement receptors in the bone marrow, and now you can have B cells recognizing that. They also start to express CD25, which is the IL-2 receptor. IL-2 is typically thought of as a T cell growth factor, but it actually will induce proliferation in lymphocytes. Well, where do you have proliferation? At the large pre-B stage, right? That's where you want to make those cells proliferate, so that's why it's expressed there. Okay, so that's a lot of information, but when you talk about what did we just cover? Well, you got to think about B cells are rearranging their gene segments to generate a new BCR so that you can have an immense diversity. And given current estimates of the potential diversity of the number of BCR you can generate is about 10 to the 23rd, which now you're talking about, you're getting close to Avogadro's number of different B cell receptors. That's way more than the number of different things that could potentially infect you. A human has about 10 to the 12th B cells. So the potential diversity is a million times bigger than the number of T cell or number of B cells you have. And so if we were to look at two genetically identical twins, they would have virtually completely different B cell receptor sequences. So this is the process by which you're generating enough diversity that you can recognize anything that comes into your system. Now if we think about this, the heavy chain and light chain are timed and coordinated. So an easy test question is, how many times does a B cell at minimum undergo somatic recombination? What's the minimum number that has to happen? Anybody? How many times does it do it for the heavy chain?
Twice?
Twice. And for the light chain?
Just once.
Okay, so at a minimum, you have to have 3 somatic recombination events. Can it have more than that?
Yeah, if it fails the test, then it has to go back and redo it, right?
Correct. So you could have a B cell could do 3, it could do 23 times. But eventually you're going to get a B cell that's expressing a B cell receptor.
I guess this is a bigger question, but why doesn't the cell just die after that? And instead of going back, it seems like going back could cause more problems. Maybe that's not true though.
I think it's a conservation of resources. So you've spent a lot of energy into developing a B cell this far. Which is more conservative? Going back one step or starting all over? And a lot of it has to do with hematopoietic stem cells only sort of tick over once a month, or about every 30 days you get a hematopoietic stem cell generating all of the downstream cells. And so it's not really like happening all the time. It's just as you go down the cascade, you have lots of cells doing this, but it's really just how many steps do you want to go back? And I think going back one step is far easier than going back many steps. So what do you need to know? We are going to ask you about what are the enzymes in somatic recombination. Okay, tell me what TDT does. It adds non-templated nucleotides. What does Artemis do? Just so when you're studying for this, for the next exam, sort of draw out what the enzymes do and what order they're doing it in. Because it's important for understanding this coordinated series of events that's leading to recombination and then test. And so you should know, okay, if I say to you, what is a large pre-B? Is it rearranging the B cell receptor? The answer is no. That's undergoing division. If I say a small pre-B, what is it rearranging? Heavy chain or light chain? Well, it's rearranging the light chain. So you should know how the somatic recombination ties into the different stages of B cell development. I'm not going to ask you what CD43 does. I'm not going to ask you, tell me about CD25 expression during all of the stages of B cell development. But I might ask you, why is CD25 expressed at the large pre-B stage? Well, it's a lymphocyte growth factor. That's where you're proliferating. So of course you have to express the receptor for that. Okay, so, and obviously I would actually ask you, if I have a pro-B, does it express the heavy chain? Well, no, it's still undergoing recombination. It doesn't express that until it's being transitioned from a pro to pre-B. And then if you get an out of frame recombination, how far back can you go? So you can't go from an immature B cell all the way back to a pro B cell and rearrange the heavy chain. You only get to go back one step. And then how do you know if you're testing your cells for self-reactivity during negative selection or central tolerance, how does it know what's going to happen? Or how does it know whether it's self-reactive? Okay, any questions about that?
I had a question in regards to the development of cancers from the pro-B cell or pre-B cell, because if CD25 is a growth factor, would you see more cancers develop from a pre-B stage versus a pro-B stage?
So you do get cancers from all of these different stages. And so myelomas and lymphomas, among others, are some of the most common forms of cancer. And they arise from this process, primarily, where you're recombining the DNA, and it's a dangerous process, okay? So yes, we try and regulate this as best we can, but you do get cancers from all of those different stages because you've turned on the recombination machinery during both the pro and the pre-B stage. You can also turn it on after you've gotten to the immature stage, but you're basically going back one step. What we haven't talked about is somatic hypermutation, which occurs after a mature B cell has been activated, but a lot of the same enzymes participate in that. And so myelomas typically are from mature B cells that have undergone all of these processes and then been activated later, where other lymphomas are these developing, pro B cell and pre B cell lymphomas. They can occur at any stage of B cell life. Any other questions before we talk about the bonus question?
Quick question. The CD25 is only with the large pre-B because it's the one that's undergoing cell division.
Correct.
Okay.
No, we say that it doesn't just instantly shut off, it sort of trails, but the highest expression is at that stage. I'm just going through the chats to see if there's any questions. The question was, can I post those middle slides? The answer is yes. I just didn't want them to have you have those before class because that was supposed to be the in-class exercise. Okay, well, if there's no more questions, let's go to the bonus question. So, Bruton's tyrosine kinase is the signaling protein immediately downstream of the Ig alpha Ig beta complex, right? It has the ITAMs that a developing B cell will use to test whether or not there's something there. Now, patients that don't have Bruton's tyrosine kinase have no mature B cells. In fact, this is a B negative, T positive SCID. And it is the most common form of that kind of severe combined immunodeficiency, where patients just don't have any B cells. And they become very susceptible to bacterial infections. If you were to look at the precursor B cells, where would they all get stuck at? What stage of development would they not be able to pass? And this is defining for patients with this particular deficiency. Okay, so that will be up on Canvas within minutes for you to be able to answer, and that gives you an extra point on the final exam. Again, you don't have to be right, you just have to make a good effort. And I'll stay on for a couple more minutes in case there are any other questions.
Because this is kind of a broad question. If if people have reoccurring infections and they're constantly sick, for some reason, maybe it's a genetic reason, or they're taking an immunosuppressant, and they're really pushing their immune system um, to be able to respond to these infections, has there been evidence to suggest that they develop cancers related to the immune system more so than other people?
So there are a number of immunodeficiencies that instead of protecting you, you are more prone to cancers. And these typically happen at developmental steps. So for example, the proteins Icaros and Notch are involved in determining, do you become a lymphocyte or do you become a T cell? And if you don't have those proteins, what happens is you get sort of abnormal T cells that are more prone to becoming cancers because the system's trying to force things through. The other way that that can happen is that if you get constant inflammation and constant responses, then you're going to be more likely to get cancer because that inflammation can lead to DNA damage. So people who have more infections typically do also have more problems with development of different types of cancers. And I would say if you're interested in that kind of thing, that's precisely the types of patients that we diagnose in the advanced immunology class.
Are there case reports of like someone battling an infection and then they develop a lymphoma a few months later, and is there evidence to suggest that, oh, you might have developed that lymphoma because of that active infection you were fighting?
It's pretty hard to tie those because you have to tie them
Right.
epidemiologically. But we do know that certainly there are viral infections that are associated with different kinds of cancer, right? HPV and cervical cancer. There are different herpes viruses that are associated with different kinds of lymphomas, but those are mainly because EBV and cytomegalovirus will infect B cells and cause their chronic activation. But even, I think the easiest example is HIV, where you start depleting your immune system and you can get things like Kaposi sarcoma, which is a rare sarcoma of the skin that doesn't occur except in a very select group of fairly consanguineous Middle Eastern men. And it doesn't occur anywhere else except now it shows up in HIV patients. But it's really hard to tie specific infections to specific types of cancers just because a one-off, there's no way to prove it.
Dr. Blotman,
Yes.
so earlier you were mentioning that the HVR3 is the most diverse area and it's at the very tip.
Well, all
Can,
three are at the tip of at the protein level. All three are the fingers from the immunoglobulin taco.
okay. Can you technically modify just the HVR3 area? Or would you have to address all of like HVR1 and 2 as well, since they're all in that same location?
What do you mean by modify?
Like, could you go in and make that antibody specific for something else, or?
Well, to make it specific for something else, you could modify any of the three, or any of the six really, right? On the heavy chain or the light chain.
Mm-hmm.
If you change any of those, it's going to change technically what it binds to.
But you would have to do all three for it to be effective, or can you only do just one part of it?
Well, if you change any one part, it's going to be specific for something else.
Oh, I see what you're saying.
But to make it intentionally specific for something you want it to recognize, you'd have to modify all six.
Gotcha.
Okay, if there's no more questions, we'll see you on Tuesday.
Thank you.